Or she gets violated. She's like,
Okay. So cell migration. There's gonna be a lot of stuff outside the textbook for this and next lecture, in part to update it and make you think about concepts and so on. So I start with this movie to basically show you a lot of the lecture is going to be about single cell migration, although there's a great deal of collective cell migrations out there. But yeah, it's going to be simpler to focus on these guys. So if you look at this one, what do you see?
migration.
Yeah, sure. So you see ruffles, right? Good. What else? Is it going in some? Does it keep changing direction? No. Why is that? So do you see a front and a back clearly? Yeah. So what is that called? Polarity, right? So polarity, ruffles. Eventually, there's move. Is that cell leaving stuff behind? Not so much, right? So all of this stuff you can basically break down the process of cell migration into simpler units, right? And that's what we're going to do, and we're going to see how it connects a lot of the biology we've talked about so far, right? So indeed, if now you take snapshots and you can decompose the whole cell migration movements into individual processes. So the first one you saw had to do with the cell orienting with regards to the direction of movement. It sort of knows where it's going to go before it's going. Hopefully, that's true for all of you when you're going somewhere. I know it's not always the case. There's wandering, but sometimes you know where you're going. And as we saw last time, this involves the PAR complex, right? And in migrating cells, the PAR complex tends to localize at the front or the leading edge of the cells, right? And there's a role of one of the GTPases, which we are going to see over and over again. That small GTPase CDC42, which contributes to that positioning, right? So now, then, once the cell knows where it's going, one of the first thing it's going to do, and you've seen that, is extend the protrusion. So there are different kinds of protrusions. They are usually actin-based. So you have underlying actin. We're going to see that again. And so here, for example, is a lamellipodium. And when we're going to see different ones, I'm going to highlight them. But so lamellipodium means it's a rather flat sheet. And the paradigm here is a cell migrating on one substrate, right? So it's kind of so-called two-dimensional migration as opposed to 3D, which exists, and we're not going to talk about much today. Right, so then once, an interesting analogy actually for cell migration is the analogy of a rock climber or someone who's going to crawl on the floor. But a rock climber is kind of nice because you need to know where you're going. You extend a protrusion, and then you need to grab on something. Okay, otherwise you have no way. The force you might exert is not going to translate into movement. So that means creation of adhesions. And so although we had to skip the cell, and cell matrix adhesion lecture, I'm going to touch upon cell matrix adhesion at the end of this lecture. Right, so creating an adhesion such that now the cell can indeed move forward. Right, it's sort of stabilizing that protrusive activity. Otherwise, these lamellipodia or filopodia, they can be formed and retract and form and retract. But to stabilize them, you need adhesion. And then you can finally translocate or move the cell body. So usually by cell body, we mean the kind of thicker part where the nucleus is. So this would be the translocation step. And it often does come together with the fact that, like I showed you, the cell is not leaving much behind. It can happen, but in this case, it doesn't. And so you have de-adhesion at the back. So I don't know why this all happened at once. old adhesion. And one phenomenon which I'm going to highlight here, although I'm not going to talk about it much today, is this concept of recycling adhesions, right? So for example, right, I showed you create adhesion here, you're removing adhesion in the back. And all of this involves integrins and complex machinery. So cells, migrating cells, often have one way to recycle these adhesion machinery, right? That's depicted here. So what do you think this would involve? Retrograde something? Yeah, to some extent, so you're on a good track. Vesicles, maybe? Where would these vesicles come from? Endocytosis of, you said that? I didn't hear it. Endocytosis of old adhesion and recycling towards the leading edge. Okay, so again, all of these lectures are connected. Now, of course, there's a strong mechanical aspect of cell migration, and this involves definitely the cytoskeleton. So we're going to talk quite a bit about the cytoskeleton today, mostly the actin cytoskeleton, but you can basically take home that all the components are involved. The actin is the most dynamic, is the one responsible for creating these lamellipodia. But there's also addition of membrane and microtubules are involved, and vesicle trafficking is involved, right? So the whole thing is rather complex. Again, cell migration is a very integrative process. And we're going to see again briefly the role of the different kinds of actin cytoskeleton here. So basically today's lecture has some slides that are sort of very general from the textbook and others that are directly from research articles, and that's what I'm going to try to emphasize today. Basically, the spirit of the lecture is now you're more grown-ups as scientists, so textbook stuff was fine at the beginning of the course. Now we're going to dwell a little bit more into research articles and actual research. All right, so briefly, just to help you guys connect the dots, we talked about the APKC PAR complex. I just said that it's localized here at the tip of the leading edge, and it's involved in polarizing all kinds of cells, right? We saw the example of C. elegans, but it's active in migratory cells, in epithelia, in the fly oocyte, neurons, and so on. The core components of the cytoskeletons are the ones we talked about. So to understand migration, you can't have forgotten everything about actin dynamics and the gaps and the regulators of the regulators that control the distribution of the actin cytoskeleton. That's what I'm going to talk mostly about. But of course, these other guys are also involved. So I'm going quickly through these very general slides we've seen before. Again, one concept which I've tried to drive home over and over again is the idea that the actin cytoskeleton is found throughout the cell, but it's present in different pools, and the properties of each pool is different, right? So at the front, you have very dynamic meshwork, you have phylopodia. At the back, you have more stable, long stress fibers, and so on. And this is regulated very locally. I'm going to illustrate that, better with this movie of a crawling cell. And hopefully you can appreciate the fact that, so you see, so that would be the leading edge. So you have at least two different kinds. It's going to play, and I'm going to explain what is there to see. You have at least two different kinds of actin dynamics. So you have these rather bright bundles. They are embedded in the lamellipodium, but they are bright. So those are bundles of actin filaments. They basically, they are akin to phylopodia. There's very tiny phylopodia. And like I said, they are embedded in this more diffuse staining here. So that's now a meshwork of actin. Okay, so thinking about the actin lecture, elongated filaments bundled together. They have some proteins for that. the actin meshwork, you have ARP2/3 complex, CDC42 rack acting there. More in the back, you see these bright foci, right? And as the cell moves forward, you see they come and go in some ways. So those, it may not be trivial for you to know that, but they are associated with the adhesion complexes. Okay? One of the key aspects of today's lecture that I'm going to finish with at the end of the lecture is the idea that when you have adhesion, when the cell adheres to the matrix, you have to have a mechanical coupling with the rest of the cytoskeleton. So usually, wherever there is cell matrix adhesion, very quickly there is also a high concentration of actin and a coupling mechanism. Okay, otherwise, there's no way the cell can move if it's not mechanically coupled to the substrate. Okay, so when you try to run on ice and not going anywhere, that's kind of the idea. Okay. And the last point I want to highlight, so you see there's a lot to unpack in just a simple movie like this one, is here. So you see at the back, you have much more stable filaments. There's not so much dynamics. Those filaments are a bit longer as well. So they are, again, themselves stable over time. They are not being polymerized and depolymerized. And yet the cell is kind of squeezing forward. So what do you think is responsible for that? Myosin, right? So indeed, you have the so-called actomyosin contractility basically squeezing the tail forward. So again, repeating myself a lot, you have these, at the subcellular level, various kinds of types of organization of the actin cytoskeleton, right? I just showed you this filopodia here, the lamellipodia, the leading edge, and the cell cortex with some stress fibers. So now what do you think can actually organize the actin cytoskeleton in such exquisite subcellular spatial pattern? The power complex is going to be part of it. It's going to help the cell know what's at the front. But what's more directly controlling actin organization? Yes?
Filament and fibrin.
So the filament and the fibrins, yes, so they will be almost effectors, if you will, to keep the filaments in some ways together. Now I'm thinking more about the control, like the regulators, the Rho GTPases. You knew that? Why don't you say it? You gotta speak out louder. Okay, or raise your hand. Okay, very good. Yeah, the Rho GTPase, right? So, well, before I show you again the most important slide, the whole idea, and I'm going to illustrate that, is that you have to have local and subcellular cytoskeletal organization, right? So something special like lamellipodia in the front, stress fibers in the back, and so on. You're going to use, the cell is going to use special signaling pathways that act very locally, okay? And so, actually, let me use this slide to illustrate an important point. I'm going to make a comment about why I want to talk to you about optogenetics on Wednesday. Because these are the kinds of tools that we need to implement if we want to study the activity of these molecules at the appropriate spatial and temporal scale. What do I mean by that? If you need a protein to work over here, but not over, and have one job over here and a different job over there, and so on and so forth, there's no way you can learn about these subtle spatial subcellular patterns with a mutant, right? Because a mutant removes the protein from every place. So in other words, the whole point of me talking to you about optogenetics on Wednesday is to show you now new approaches where you can now start to interrogate the function of proteins at the time and place where they really work, okay? With the appropriate spatial temporal resolution. Okay, so indeed, the Rho GTPases are key regulators of the actin cytoskeleton. We saw that, so this is just a brief reminder there might be an excess held in the cytosol by this GDI, guanosine dissociation inhibitor protein. The Rho GTPase need to be anchored at the membrane to function. So this is achieved through lipid modification, so addition of fatty acids at the C-terminus usually. And but still, see, if they are still bound to GDP, they are inactive. That's the inactive form. They are activated upon interacting with the GEF, or guanine nucleotide exchange factor. That is one part that's in response to signaling. I should emphasize here that signaling can also modulate and help recruit gaps. Okay, so you can have both activation and inhibition of Rho GTPases being controlled locally by signals. And then, as you know now, I hope you're getting bored because feeling like, oh, we know that, right? Am I? Yes? Okay, good. I want you to be a little bit bored, quite frankly. I mean, because it would be a good sign, that's my point. So, right, once they are in the GTP-bound state, the conformation is such that now they can interact with the effectors. Now, let me just use this slide to illustrate two tools I'm about to tell you about. One of them is the concept of constitutively active. Actually, let me ask you this. If you had to design a constitutively active GTPase, a GTPase that cannot be inhibited by gaps, what kind of mutant should this be? Yes?
Make it non-hydrolyzable.
Yes.
GTP.
Let me rephrase this slightly, making it unable to hydrolyze GTP. That's exactly right. That's exactly right. So there are mutants, usually point mutations, so a single amino acid is changed, and now the GTPase is unable, like you say, to hydrolyze GTP. So it's now in a constitutively active form. Okay? And so that's the CA, or constitutively active, as if they are stuck in this form. And there are other kinds called dominant negatives. So those are unable to let the GEF kick out the GDP. In other words, they are constitutively bound to GDP, but they still interact with the GEF. So what they do, when you overexpress such a dominant negative, they compete with the endogenous GTPase for the GEF, and they block activation of the endogenous GTPase. They're not very efficient, they're not very good tools, but they still kind of work to inhibit the function of Rho GTPases. And I thought you needed to hear that because a few of the experiments I'm about to show you involve these tools. So let me briefly go over kind of a very traditional assay to study cell migration, the so-called scratch assay. So what people were doing is use cell culture, so-called confluence. That means they cover the entire surface of the Petri dish. And then with a small cover slip or knife, induce a scratch, which can be seen in this way. So you have a whole lot of cells here, and there's a scratch has been made, and after 5 minutes, it's still empty, but very quickly, well, within a few hours, cells actually crawling. Okay, so and this was used as a paradigm for cell migration for a long time because the distance is greater than a single cell diameter, so cell division does not account for filling the gap. It's instead cell movements. Okay, so in cartoon form, so they move into the wound. And that's it. So basically, by monitoring how quickly, how efficiently the wound can be healed, if you will. So healing is a big word here. Healed. People could get use this assay as a proxy to study cell migration. And so in this case, so these are very ancient studies, but they led to the working paradigm, which is still present in textbook and so on. So people were overexpressing now constitutively active or dominant negative form of the small Rho GTPases, right? So to either block their activity or force their activity everywhere. And so sometimes, so the ones I call constitutively active sometimes are called dominant active. but that's the same idea. So here, overexpression of constitutively active forms of Rho, RAC, and CDC42. So the human genome and many vertebrate genomes have a lot more GTPases, like 17. But those were the three that have been the most studied and that seem to be doing a whole lot of activity. So you see here, you have, actually, maybe you could think about that. So what do you see? Especially if you compare to the images I showed you before. Stress fibers, right? It's like the cell is filled with stress fibers, right? Now with RAC, all of this is actin, I haven't said. This staining here is actin. So you have this dense meshwork at the periphery, more lamellipodia type. And then with CDC42, you get this crazy production of filopodia all over the place. And if you remember, the cell that was left on its own device to migrate had all of these things. Stress fibers in the back, lamellipodia and filopodia at the front. Okay? Now, if you inhibit the function of either of these three GTPases and you quantify the wound closure in the scratch assay, all of them or interfering with the function of any one of them causes a reduction in the ability of the culture to fill the gap. Okay, so that suggests they are all required for cell migration. Okay, so now if you would put this data together, the idea would be, well, in the cell where you have filopodia and lamellipodia at the front, that's more like a RAC and CDC42 action. And in the back, stress fibers, it may be more of a Rho activity, right? So this is the kind of study that led to this model where in terms of function, and so this model is still largely, there's a great deal of truth to it. Okay, so at the front, upon sensing some signaling via growth factor and whatnot, or a gap, actually, sorry, when I say gap here, I mean a hole. So in the scratch assay, because you should be wondering, why are they moving? Well, it's because they sense that they don't have a neighbor anymore, and they have a neighbor on the other side. So that's the extrinsic information that tells them to polarize and start moving towards the gap. Okay. And so it's not the same gap, right? Okay. So CDC42 RAC, we saw this. They can both activate ARP23. And so what does ARP23 do? Branching, right? It comes to the side and helps nucleate de novo, but from a branch. And indeed, but still, it's kind of, they both activate ARP23, but somehow this guy makes filopodia, this guy makes lamellipodia. So to me as a student, I was never really satisfied by this kind of diagrams. I'm like, okay, we're missing a whole lot of information. So we're going to see some of that today. But on the other hand, we have Rho A, which can do two things. One of them we've seen before, right? It can activate ROCK or Rho kinase, which phosphorylates myosin light chain, activates myosin. Where did we see that? Polarity, yeah, in the C. elegans cortex. Okay, so that's true in many, many contexts. Almost every myosin activity, except in the muscles, is regulated downstream of Rho in this way. But Rho can activate other effectors, including this formin. Where did we see formin? Actin. Oh yeah, actin polymerization. Yeah, sure, it's written right there. Yes, but so this was the one that helps nucleate and then sits at the plus end and promotes, blocks capping and promotes elongation. So I want to emphasize this one because these are, in some ways, these are two almost antagonistic functions of Rho, right? But then, in spite of, so together, people interpreted this as, okay, it promotes the formation of these stress fibers and the myosin activity that's going to use those as a substrate. So that's for the activities, and for the subcellular localization, the model was that you have CDC42 at the front, and that makes sense also with regards to the PAR complex, right? Because I showed you the PAR complex is at the front, and the PAR complex actually contains CDC42. So these proteins interact with CDC42. So here you have potentially already a connection between the polarity, so where the PAR complex is, and CDC42 activity, and then the activity of the cytoskeleton. Okay? And so, and one idea is that indeed at the front between the two, CDC42 and RAC, they help promote this lamellipodia formation with some filopodia, and somehow help activate, or there's some antagonism between the two that keeps Rho activity in the back. Okay, so now what we're going to do is we're going to reevaluate this model a little bit with more modern techniques. So one such technique is so-called FRET. Have you heard of FRET before, or fluorescence resonance energy transfer? Yes, I see nodding people, so that's good. That's a good sign. Let me just give you a quick reminder and also explain how it's been used in this context. So you remember that Rho GTPases work by interacting, so physical protein-protein interactions with effectors. Okay, so when you have in cells, so if we look at this example here, when you have active RAC, so bound to GTP, now it can bind to this PBD. So it's a binding domain for RAC. Okay, and if you engineer these proteins in such a way that RAC has GFP, but the PBD has a red fluorescent molecule, or RFP, for example, or at the beginning, people use the synthetic compounds. Then if the two are close enough, you can get FRET, right? So you know that FRET is when you activate a fluorophore that will absorb higher energy, so lower wavelength. If there's another fluorophore that is compatible to basically get excited at slightly higher wavelengths, so lower energy, FRET can occur. In other words, there won't be emission of light, but the energy will be passed on through resonance to the neighboring protein. So this is a proxy for measuring proximity. Okay, so this principle was used in this first generation of FRET sensor. You had then another generation, so this is now getting old, of so-called unimolecular FRET sensors, where now you have the two, so the Rho binding domain and the Rho GTPase on the same molecule with YFP and CFP. And when the Rho is inactive, there's no binding to the RBD here, so the level of FRET is low. There's always some. It's low, and when you have activation, now this protein is folding onto himself, bringing the YFP and the CFP very close together. Now you can have higher FRET, right? So, for example, if you look at these kinds of measurements, which you can look at, when you study, but basically, so here would be two constitutively active form of Rho. So this is an arbitrary measure of FRET. So that's the maximum, let's say. And this one here is a dominant negative form of Rho. So it cannot bind. Okay. So here the difference between the two would be your so-called dynamic range, right? Because this one is the baseline, is the amount of FRET you get when the protein is constitutively in this inactive conformation. And that's just because this mutant here is a dominant, is an inactive. You see, if you inhibit Rho with a gap or with GDI, you reduce this FRET activity. Okay? So this is just to show you that people could engineer molecules to basically monitor the activity of Rho GTPases through FRET. Now, FRET, that's a microscopy-based measurement. So here I'm showing you bulk measurements, but that means you can now measure this in cells. Okay, so now this is getting really interesting, right? Come on. Because now you can, so these are live cells, and this color code scale bar is showing you the amount of fret. So that's a proxy for the activity of these row GTPAs, in this case, row A. So you see now you have a cell migrating, that's the cell body, it's migrating in this direction, it's migrating in this direction. And now you start seeing a bit of something that should surprise you. Because here this is the highest, and that's row A. What did I say about row A? It's active in the back. Well, that's what's showed here. Indeed, there's a lot of activity in the back of the cell. But on these cells, there's a lot of activity in the front. Right? So it's not so simple. Okay, so let's try to look into this. So, indeed, so here is a cell that's going to be stimulated by PDGF, and by default you have this line very close to the leading edge, very close to the membrane, and before treatment you have a certain activity, that's what's shown here in blue, very high near the leading edge. So this is the distance, so basically here that's the leading edge, and this is further into the cell. Let's say if you were to have a cross section here. And then people treat with PDGF. What you see here is what you see here. You see a reduction of row activity. So now you start getting some insights into the dynamics because you can watch the activity in live cells. That row is almost by default at the very tip, very close to the membrane at the leading edge. But upon treatment, and that's a treatment that's going to stimulate migration, that's going to stimulate protrusive activity, you have a reduction. And the interpretation actually was that, so PDGF indirectly inhibits row A, in part by activating RAC, right? And you should always think the way it activates it is through a gap. So how do you think RAC can inhibit row A? Through a gap. So when I say it's indirect here, you should be able to propose the hypothesis, well, that's probably a gap, and etc., etc. Okay, so that's fun, but again, here cells are being treated with the chemical that's present in the medium throughout. So we can do better than this. We can start using, I'm going to start teasing you with optogenetics, and then I'm going to tell you more about this on Wednesday. So some of the first optogenetic tools to manipulate the activity of row GTPases were in this way. So now, right, the hypothesis is RAC inhibits row A. Can we, more or less indirectly, can we test that? So here the tool is the following. So you take a RAC protein, and you have to know that this has to be a constitutively active RAC. Okay, so inherently it's going to activate if you let it bind to the effectors. Except it will not bind to the effector because itself, so this is entirely engineered, right? It doesn't exist in the wild, but this RAC protein has been attached to two domains from a plant protein, which basically interact with each other, and somehow it folds onto itself. In other words, it's auto-inhibited. Okay, so the presence of these domains, of this love domain, inhibits RAC activity. But then when you shine light of a given wavelength, this association breaks apart. Now it's more loosely connected, and now RAC can interact with the effector. So what you've basically done is, by shining light on the system, is activating RAC. Okay? It's been called a photoactivatable form of RAC. So let's do that. So here's a cell, and what you're going to see is that people are going to, every time you're going to see a circle appear, that means some light has been shone onto this part, and trying to activate RAC. So, alors. Okay, so this is happening here. So the cell is laying flat on the substrate. Boom. First one, so you see there's some ruffle. You see, and it kind of tends to contract. And they have to do it several times, and eventually it starts sticking up. Okay, but you see the whole area is moving. Okay. Right, so that's in summary what they've done, and indeed you activate RAC for a little while, and then eventually the cell starts protruding. Now, is it RAC that will end up inhibiting Rho A? So now you can do really interesting experiments where on one hand you're going to photo manipulate RAC activity, and on the other you can monitor Rho A activity. Right? So we're after the question is really RAC inhibiting Rho A locally. So in this experiment, in the control, so this is now we're using a Rho A biosensor based on FRET. So you see a whole lot of activity at the leading edge, and this is the axis onto which this was measured. And now you locally activate RAC, and sure enough, in the control you have high levels of Rho A activity near the leading edge, but when you activate RAC, it goes down. Okay, so we're now starting to dissect these interactions between Rho GTPases in a very local manner. Okay, so that means this model, initially we kind of have to refine it because the model was saying PA, so RAC activates Rho A, but somehow activates it at the back. If you look back a few slides ago. Now we're saying, well, locally RAC inhibits Rho A. So let me show you, I'm going to show you a very complex analysis that I'm not going to ask you to sort of memorize because it's very complex, but it's very important to illustrate some new paradigms. So, like I showed you, so this is in this paper, which is now 10 years old. Again, so this was monitoring in live cells the activity of RAC with FRET. But in this paper, what they could do is monitor the activity of all the GTPases. Okay, well, the main three: RAC, CDC42, and Rho A. Okay, so that's the kind of measurement they do. And then they do a really clever type of analysis. So here you see, right, measurements for RAC, measurements for CDC42, measurements for Rho A. And what they're really trying to ask is, what is the real sequence of events leading to the formation of a protrusion, right? And again, the reason why I'm emphasizing protrusion is the first step in cell migration, right? To make, and we know this is controlled by these Rho GTPases. So what they do is create this, film these cells and get these movies, and then segment the cells in small areas like this, the so-called sampling windows. And so sampling windows either very close to the membrane or further inside the cell. And for each sampling window, so you see here a whole lot of sampling windows. For each sampling window, they quantify RAC, CDC42, Rho A, and the protrusive activity, right, in time. So when they can quantify all of this, they can start asking, and this is a convoluted analysis, so try to understand, but I'm not going to bother you with that. They are looking for correlation, so it's a so-called cross-correlation analysis. In other words, they are trying to ask which activity of Rho GTPase correlates the best with protrusive activity over time. Okay, so what this is showing you here is every row here is one of these windows. Okay, and the zeros is when that particular window was protruding. Okay, so that's the time of protrusion. That's the phenomenon we're interested in understanding. And so they see they have this value for many windows. And now they ask, in terms of RAC1 over time, so either before the protrusion or after the protrusion, when is the best correlation? So you see here, it's negative values means the best correlation is between the protrusion and the rack activity is slightly after. Same thing for CDC42. You have these ones here, these little windows. The best correlation between protrusion and CDC42 activity is after the protrusion. But for row A, the best correlation almost invariably is smack at the time of protrusion. Okay? So just by correlation, there seems to be a stronger connection, a stronger correlation between the time of protrusive activity and the time of row A activity. Okay? So this is summarized here in these kinds of plot. Again, here, this is the aggregated time at which those windows protrude. Row A tends to peak in correlation right at the time of protrusion. RAC and CDC42, here in green and red, slightly after. And there's almost a systematic time lag between row A activity, which comes a few, up to 50 seconds before the peak of CDC42 activity. Right? So it kind of challenges the model that protrusions were triggered by CDC42 and RAC. Because here it's showing that protrusion precede CDC42 and RAC, and instead correlate better with row A activity, which was thought to be completely absent from protrusive activity. Okay? So the new model that these guys propose is that you have, and we've seen this movie where there is this kind of ruffles and some protrusion and retraction before there's an actual So what these guys propose is that row A activity very near the plasma membrane at the tip is actually responsible for the onset of the protrusion. And then with the lag, you have actually kind of a retraction when CDC42 and RAC kick in. And remember, RAC helps inhibit row A. But then those have a longer duration of activity, especially RAC, and now this promotes the lamella. Okay, so in terms that, I know this is complex, but it's just to give you a feel for, what's going on. Now, if we look back and we try to put this information back on the textbook model, so here is how it goes, is that the idea is that the onset of protrusion, of protrusive activity, is actually triggered by actin polymerization and by elongation of filaments, by creation of filaments de novo. So that's the job of forming. That's something that can be powered by row A. Okay? But then once you create the first few filaments, now you have a template where you can start adding these OP23 and new filaments on the side. Right? And that's the job of CDC42 of RAC. And hence the idea that you start by pushing the membrane with formin, with row A and formin, creating new filaments. And now that serves as a template to create the meshwork that will power the lamella to go on. All right, so let me move on to the next part I wanted to tell you about. And that's because everything we've seen so far has to do with the making of protrusions. But we have now to connect this with the polarity. There is some connection between CDC42 and the PARC complex, but also with the signaling. So I'm going to give you a very classic example of this. Very thirsty today, somehow. All right, so this is a dictyostelium. It's a little eukaryote. And this is a pipette tip. And with this pipette tip, what people are doing is delivering cyclic AMP and seeing where this dictyostelium goes. So they kind of mess with the dictyostelium by moving the pipette tip. It's as if I was giving you an exam and you were going there and then I'm saying, no, now the question is this. And then you answer, and no, now the question is this. It's very tempting. So basically, what this is showing you is fairly remarkable, right? Is this cell, that's a single cell. It's not like your cat or something.
How long is that? Is this like?
Well, this is like real time.
Wow.
Yeah, so those cells, so you have cells, and in the body you have neutrophils, human neutrophils. They are specialized in chemotaxis in this way, so they can move very quickly. You can respond to the gradient of concentration of a compound like here CMP, and they can reorient with regards to that gradient and move towards the source. Okay, so that's true for dictyostelium responding to CMP. It's true for human neutrophils responding to FMLP or formulated and whatever this thing is coming from bacteria, right? So neutrophil will hunt bacteria down in the same way. And if bacteria tries to escape, neutrophil is going to chase them. I mean, there are movies out there showing that. So this was shown in both cases to rely on signaling from a lipid membrane. So now that should throw you back to one of the early lectures, right? Membrane lipids, so these fatty acid chains and this glycerol residue and this inositol here. And this inositol can be more or less phosphorylated because it has these hydroxyl groups. And you have enzymes dedicated to controlling that. Kinases will add phosphates, of course, and phosphatases will remove phosphates, so this P10. And what's been determined is that it's the local concentration of certain form of this phosphoinositol that determine the polarity and the direction of movement for dictyostelium and for neutrophils. So for example, here you have this PI3 kinase is very important to go from PIP to this PI3-4 diphosphate, or it's one form of PIP2. Okay, and P10 does the opposite. So if you look at P10, the phosphatase, a classic picture here is that it's present throughout the cell at the cortex, but it's particularly devoid at the leading edge. This cell would be migrating in this way. Okay, so everywhere in the cell, but at the leading edge, you have the phosphatase that dephosphorylates this particular membrane lipid into this form, right? I know it's complex. I see a lot of... That's fair. I was the same. So my point is, there's a whole suite of proteins to regulate the local concentrations of these compounds at the membrane, right? So it happens at the membrane, which is where you want things to happen to integrate signaling and respond with protrusive activity. So indeed, P10 in the dictyostelium, when you put CMP on one direction, P10 tends to accumulate at the back. Phosphokinase, the lipid kinase, PI3 kinase tends to accumulate at the front in addition to its accumulation in the cytosol. Right, so basically you could explain a gradient of PIP2 at the membrane simply by positioning P10, the phosphatase, in the back and the kinase in the front. So are we getting such a gradient in the cell? One way to visualize that, and that's a busy slide, but actually I only want you to get one aspect of it. So it's this so-called pH domain, or plextrin homology domain here. That's something people have used. So they have basically taken this domain from another protein here, this protein kinase B. And the reason why you want to do that is because it will bind specifically to this PI3-4 biphosphate. So now if you take this domain and you attach it to GFP, you now have a sensor for the concentration of P2. Okay? And here is what you get indeed. So if it's not the same cell. That cell would be bigger, but you see much higher concentration of PIP2 of this lipid at the front and P10 at the back. So it's all indicating that you have the phosphatase at the back, the kinase at the front, and you tend to accumulate the lipid at the front. Right? So if I summarize, this is what we've seen. The kinase, the phosphatase, leading to the accumulation of the lipid at the front. Now the question is, how does that relate to what we've seen before, which is the distribution, the polarization, and the activity of the actin cytoskeleton? Because we still have to explain how this leads to the polymerization of actin at the front and the actomizing contractility at the back. So let's look at this paper here. So this is not on dictyostelium, it's on neutrophils in the zebrafish. And the paradigm is that if you take a zebrafish and you have here fluorescent neutrophils. So in the movie, you'll see they crawl around, okay? And what you do is with the laser, you wound the fish in a very specific place. You're gonna see that in the movie. So you can wound the fish and you'll see the neutrophils, they run towards the wound. And they run because the wound causes secretion of NO, so natrium oxide, whatever, NO. And that serves as an attractant for those neutrophils. So let me just play the movie and then I'll tell you what this is. Oh no. Okay, look at it. It's gonna play again. So here, you see the wound is the blue dot. So see the wound is made and they literally jump to basically help take care of the wound. Okay, so they contribute to the healing. And here, you make a wound and nothing happens. But the difference between the two is this is a control. This has been treated with a drug, LY294002, which I obviously do not need you to memorize. That's an inhibitor of PI3 kinase. Okay, so basically this is a first evidence that in this paradigm, you have neutrophils that will jump in response to a wound, very directed migration, and the system needs PI3 kinase activity. Similarly to what we've seen before, in this paper, they created a reporter for PI3 kinase, and it's a very clever one. So here you have M-cherry, just to label the outline of the cell and give you a feel for whether the cell is thin or big and so on. So it's sort of normalizing the signal. And here you have pH, so the same pH domain from the protein AKT fused to GFP. So that's going to bind to PIP2, okay, the phospholipid. And if you calculate the ratio of the two, you get this signal, and that tells you very nicely where the PIP2 is. But now you can watch it live, okay? So let's see, where do you expect PIP2 in the cells? At the leading edge, as per the textbook model. Oh no, why does it always do that? Here's the wound. Well, don't worry. See, so by and large, indeed, the PIP2 activity tends to be positioned at the leading edge. Okay, so that's all consistent with what we've seen before, which is those cells chemotax towards the wound. They need PI3 kinase, and they do have accumulation of the PIP2 at the membrane towards the leading edge. And you see that's a very dynamic process, right? So this is partly how the cells can reorient so quickly, like in the movie you've shown. But still, we haven't answered, okay, what's the connection between this and the actin cytoskeleton, right? Because eventually the actin cytoskeleton is what powers the phenomenon you're seeing, that is those protrusive activity. So here what the experiment is again very clever, and that's using the same photoactivatable rack we've seen before. So the idea is, well, if the PIP2 accumulation and PI3 kinase activity somehow feeds into, for example, rack activity, is it possible to rescue the protrusive activity by supplying active rack, in the absence of PI3 kinase, right? So this is the kind of experimental design by the end of this course. So by now, yes. Oh, Sure. So that's the phospholipid. The lipid at the plasma membrane that needs to be phosphorylated or dephosphorylated by PI3 kinase. And I'm going quickly. So this is the membrane lipid. And it has this inositol group and has many hydroxide groups, many OH, and that can be phosphorylated. So it's just when you have a particular form of it with two phosphates on two specific positions, that serves as a messenger. Okay, it's part of the signaling, and that's the molecule that is synthesized by PI3 kinase and that is detected by the pH GFP. Okay, and here pH means plextrin homology. Okay, so in this experiment, they're going to use this drug again to inhibit PI3 kinase, but now they're going to also have PN-RAC, so the photoactivatable RAC, so to inhibit PI3 kinase, but also restore RAC activity at the front. What I have not showed you is when you inhibit PI3 kinase with the drug, if you look at a RAC biosensor, the activity is reduced. Okay, so here the idea, right? You block and then you photoactivate RAC in one position, and sure enough, the cell is actually moving. So that's the control. And when you do it, in treated conditions, so here you see the cell. There's the drug, so it's not very active. But then you activate RAC in this location, and you start getting restoration of the protrusive activity. But not the constriction at the back, right? So this is what they point out here, that restoring RAC was allowing restoration of the protrusion, but not the contraction at the back. So this was a kind of an interesting observation leading to the following model, which is that indeed downstream of this PI3 kinase, PIP2 or PIP3, so you have various forms as you see on the slide, you activate RAC, and that promotes actin polymerization at the front. But somehow the PIP concentration also feeds into the contractility at the back, and at the time they didn't really know how. And since it's been shown that cortical tension, so you have mechanical inputs connecting what happens at the front and what happens at the back. And what you might also wonder about is what activates PI3 kinase in the first place, so that's signaling. Okay, so for that you have to invoke the GPCR sensing the signals. All right, so I'm done with this lipid signaling, so I'm just going to finish in the last few minutes with going back and looking at the cell matrix adhesion. Right, so I was telling you at the back and here in these stress fibers, because as soon as the cell extends a protrusion, to move forward, it has to deposit anchors, it has to latch onto something. Okay, so these anchors, so we've seen what happens here at the leading edge, and those anchors often involve a more stable actin cytoskeleton, including these contractile bundles. So what this is basically representing here are actin filaments and myosins, and with the two hands like myosin 2. But the connection to the matrix is via these receptors here, integrins, which are transmembrane proteins that span the membrane. And basically, in a nutshell, so I'm going to tell you now what I'm going to illustrate throughout the next few slides, and that's the key concept, is you want something, so those integrins, on one hand, they can bind to the matrix, adhere to the substrate, so that's what happens here. On the other hand, they have to be cross-linked to the cytoskeleton. Okay, so that's what you need to get any kind of mechanical coupling between the cell, the substrate, and its own cytoskeleton. So if we look a bit more closely into that, these integrin receptors, again, they can bind on the extracellular matrix and on the cytoskeleton inside the cell. They tend to be composed of heterodimers of alpha and beta integrins. The beta, they do the bulk of the work to some extent. There's a great deal, a great variability of those. So here's just, I don't need you to memorize this, but just to illustrate that you can, with only a few different genes, so beta 1, 2, 3, and so on, and alpha and a few alphas, you can create a great diversity of heterodimers, obviously. Okay, because they are not, I don't know how to say, they are not monogamous. So beta 1 can be with all kinds of alphas and so on. And they're expressed in different cell types, and they have, depending on the heterodimer, you have preference for either collagens or fibronectin. Here I'm going to give you some example about fibronectin and so on. So at the level of the whole organism, you have a great variety of cell matrix adhesion options. And on the matrix side, indeed, you also have some diversity. So collagens, of course, are essential. Many of these compounds, they are essentially structural, so there's a lot of fibrous proteins that make this higher order structure, so that's true for the collagens. And then they integrate other proteins such as laminin, fibronectin, and so on. And the point here is that you have a pretty good match between integrins, right, so the receptors, and the composition of the extracellular matrix. So just to illustrate, here this would be fibronectin. So fibronectin, that would be part of the basal lamina, so it can bind. So now this would be the fibronectin molecule. It can bind on one hand collagen and some other extracellular matrix compounds. But on the other hand, especially through this RGD domain, this is how it binds to integrin. Right? So you have, again, this kind of modular organization. So that's for the integrin to matrix interaction. And like I said, you need to now couple this mechanically to actin. So in this slide, what you're seeing is F-actin, and very often you have, so the resolution, you can, there's already, I need to update this because there's so much better out there. But the whole idea is that you have these actin filaments here, and you have these foci of integrins. So basically, wherever you have integrin clusters forming an adhesion, you tend to recruit the actin cytoskeleton. So this is also illustrated here in this old electron microscopy. So the plasma membrane would run here, and you have a very direct connection between these fibronectin fibrils here, that's the outside of the cell, and the actin filaments on this side. So in here, this is where you would find the integrins and this adhesion complex. When I say complex, I mean it, because on this slide, so you have the extracellular matrix components, again, with so in the case of fibronectin, with this RGD that can bind either of the alpha or beta integrin, especially the beta subunit. They are each single pass transmembrane protein, and on the C-terminal side, the beta integrin can interact with talin or alpha actinin here. So the reason why I wanted to really highlight this is because they do not interact directly with the actin cytoskeleton. Right, so those are the so-called cross-linking proteins, talin, alpha actinin, and they have different mechanical properties, and they can be more or less stiff and so on. So the mechanical property of the coupling will depend on all of these compounds, right? Both the matrix, the integrin dimers, but also which cross-linking protein is mostly present here. And to make things even more complex, integrins can also signal, right? So they are not just mechanical anchors. The cell uses the integrin engagement with the matrix to signal. So here you see some examples. They can signal through PI3 kinase, right? So they can be part of that signaling loop at the front, especially if they start engaging. But they can also signal to other signaling pathways, and you'll see, or I don't think you've seen these pathways yet. But some of those can actually regulate gene expression. So there are many examples of cells adhering to a substrate through integrin, a particular substrate, and that seems like a very mechanical thing to do, but this can have an impact on gene expression and on the identity of the cell eventually. And to finish, that's going to be my slide. As a reminder, once you have established this connection between the cytoskeleton and the extracellular matrix, you can indeed use myosin as a key molecular motor to exert force and help squeeze the cell forward. And I'll stop here.